1. Unlocking 20 million years of rhino evolution, the tooth that changed everything. 2. Unlocking the secrets of ancient proteins, a new era in paleontology. 1. Unlocking 20 million years of rhino evolution, the tooth that changed everything. An astonishing revelation about rhino evolution has emerged from a fossilized tooth, encased in earth for over 20 million years. This ancient artifact is providing scientists with groundbreaking insights that could reshape our understanding of how the rhino lineage developed over millennia. Researchers at the University of York have made a significant discovery within the tooth's enamel, preserved proteins that serve as vital clues about the rhino's evolutionary journey and its divergence from other species. Unraveling the rhino family tree. Through diligent analysis of the enamel proteins found in this tooth, the research team was able to pinpoint when this specific rhino diverged from its relatives in the family Rhinocerotidae. This split is believed to have occurred somewhere between 41 and 25 million years ago, during the transitional period between the Middle Eocene and the Oligocene epochs. Perhaps even more astonishingly, their findings indicate that the two primary subfamilies of rhinos, Elasmotherinae and Rhinocerotinae, did not separate as early as previously thought. Unlike earlier conclusions suggesting their split happened during the Eocene, this new evidence suggests that the division likely occurred during the Oligocene, specifically between 34 and 22 million years ago. This revised timeline is crucial because it enhances our understanding of the processes through which various rhino species evolved, adapted, and faced environmental challenges, including extinction. Revolutionizing insights through DNA. While ancient DNA has been a resource for studying extinct species, its preservation generally lasts only up to 1 million years. However, this fossil tooth defies that boundary. The successful retrieval of enamel proteins from such an old specimen expands the timeline for molecular research by a remarkable factor of 10. This advancement is significant as it permits scientists to delve into rhino evolutionary history using molecular evidence rather than solely relying on fossilized structures. The well-preserved enamel proteins provide valuable genetic information, thereby unlocking new avenues for exploration within the field. Enamel, renowned as the body's hardest tissue, has a mineral-rich structure that preserves proteins over millions of years, especially in the cold conditions of the environment where this tooth was excavated. Ensuring authenticity of the fossil data. Discovered in the high Arctic of Canada, where permafrost remains prevalent, the fossil tooth's chilly surroundings were crucial for maintaining the integrity of the proteins within it. However, the cold environment alone wasn't sufficient. To confirm the protein's authenticity, researchers employed a technique known as chiral amino acid analysis. This method allowed them to differentiate between the original proteins from the rhino's life and any later contamination that might have occurred. By examining the degraded proteins from this tooth and comparing them with those from already studied rhino fossils, the researchers established that the material was indeed authentic. A unique preservation site. Professor Enrico Capellini, from the University of Copenhagen's Globe Institute, emphasized the exceptional nature of the fossil's location. He expressed that the Houghton crater might be an extraordinary site for paleontological studies, a biomolecular vault that protects proteins from decay over extensive geological timescales. Capellini noted that the unique environmental history of this area has created opportunities for exceptional preservation of ancient biomolecules, akin to how some locations maintain soft tissues. He advocates for increased paleontological fieldwork in similar regions worldwide, spurred by this finding. Delving deeper into our past. Dr. Mark Dickinson, a postdoctoral researcher at the University of York's Department of Chemistry, was one of the co-authors of the study. He remarked on how incredible it is that modern tools are allowing us to probe further back into history. With the enhanced understanding of ancient proteins, we can begin to ask intriguing new questions about the evolution of life on Earth. The team regards this fossil tooth as more than just a solitary find. It signals that many more ancient biomolecules might still be awaiting discovery. Shifting perspectives on rhino evolution. Fazila Munir, who analyzed the tooth during her doctoral research at York, added, "The successful analysis of ancient proteins from such a historic sample offers a new viewpoint for scientists globally who hold significant fossil collections. This critical finding aids us in piecing together our ancient past. As modern rhinos face dire threats from habitat loss, poaching, and climate change, tracing their evolutionary history is vital." Understanding how previous environmental changes influenced rhino survival or eventual decline equips us with knowledge that is essential for conserving the few remaining species today. The full findings of this groundbreaking study are available in the journal Nature. Enjoyed the insights? Subscribe to our newsletter for more engaging articles and exclusive content. Also, check out EarthSnap, a free app brought to you by Eric Rawls and Earth.com. 2. Unlocking the secrets of ancient proteins, a new era in paleontology. Unlocking the secrets of the past, ancient proteins and their impact on paleontology. Imagine discovering a hidden treasure trove of information about creatures that roamed the Earth millions of years ago. This is precisely what ancient proteins, found within fossils, offer to scientists studying long-extinct species. These proteins are not just remnants of the past, they are vital clues that can illuminate our understanding of ancient life forms. However, there's a catch: like all ancient biological molecules, proteins are subject to degradation over time. Until recently, the oldest proteins that could be reliably analyzed were approximately 4 million years old. But groundbreaking research published in the journal Nature on July 9 has changed the game. Two independent studies, one conducted by a team from Harvard University and the Smithsonian Institution, and the other led by researchers at the University of Copenhagen, have successfully extracted ancient proteins that may date back as far as 29 million years. These remarkable findings open new avenues for paleontologists, allowing them to delve into the behaviors, diets, and evolutionary paths of animals that were previously deemed too ancient for molecular analysis. This could revolutionize our understanding of how these species lived and interacted with their environments. But here's where it gets controversial. The implications of these discoveries are profound. They challenge the long-held belief that molecular tools could only be applied to relatively recent fossils. As we uncover more about these ancient proteins, we must ask ourselves, what other secrets of the past are waiting to be revealed? Join the conversation. What do you think about the potential of ancient proteins in reshaping our understanding of paleontology? Are there ethical considerations we should keep in mind as we explore these ancient remnants? Share your thoughts in the comments below.